All right. Hello, good morning. My name is Sonia Nesser. My pronouns are she, her, and I'm here to talk to you about research in chemistry. For the past year, I've been working in a chemistry research lab at the U of O, the Wong Lab. And I just want to tell you all about what I've been doing and just generally what chemistry research looks like, because I know when you're studying classes, it can seem a bit vague as to what research might actually look like. Um, so what is the Wong Lab research? Most chemistry labs or all chemistry labs have kind of an overarching theme. So you might have a lab that researches like biochemistry. For the Wong Lab, we research physical chemistry and materials science. So physical chemistry, very generally, is about how matter behaves on a molecular and atomic level, and also how chemical reactions occur. And then materials science is relating the microstructure of a material to its physical and chemical properties. So materials science is studying a material and then saying, okay, we need a material that has these qualities. Maybe it's a packaging that biodegrades. And how do we take what we know about just all of the materials that exist and make what we need? So how do we do that though? We use a technique called spectroscopy, which is using light to study the properties of materials. So we look specifically light, we look at two systems using a high-powered laser. We look at nanocrystals, and then we also look at organic thin films. And I'm going to be talking mostly about the nanocrystals just because that's what I've done personally. But then I'll touch briefly on the organic thin films just because it's really interesting. But first, let's talk about the laser. So over here, you can see a picture of the laser, and it's kind of cool. The laser beam is actually visible in this photo. It's not normally visible in photos, but when there's kind of particulate matter in the air, when there's, say, condensation or maybe smoke or just water droplets or dust, then you can actually see the laser, and it's pretty cool. So you can see here it comes out of this black box, bounces off that mirror, this mirror over there, and then goes to this thing, which is called a periscope. And so that means the laser bounces in and down and out. And so it goes in here, down, and then out there. And so why am I talking about a laser suddenly? I thought we were talking about chemistry, right? The laser is really, really important to the chemistry work that we do. And we wouldn't be able to do all of the chemistry studying that we do without the laser. It's the most important thing about the lab. So what is it? We use two laser spectroscopy, which we use to study how material changes over time. So the two lasers that we use have different purposes, and they're the pump beam and the probe beam. And so the pump beam's job is to send energy into the material, and that's the green beam that you can see in this picture. And the probe beam's job is to measure how that energy is released by the material, and it's a white beam that's not in this picture. So to give an analogy, say you're walking along the sidewalk and you see something and you're not entirely sure what it is. So you might poke it with a stick very gently. The stick would be the pump beam, just sending energy in, seeing how it's going to respond. And then the probe beam would be, after you poked it with a stick, you've got to watch to see how it responds, right? Because if it doesn't move or growls at you, that's two very different conclusions to what it was that you just prodded, you know? And so that kind of is roughly how our two laser spectroscopy setup works, with the poking the stick and then watching to see how whatever we poked responds. And so we put our sample, which is either a slide, so like a microscope. You all worked with microscope slides, presumably before, those little glass kind of rectangular slides, very thin. So we either put that or a cuvette, which is two very thin layers of glass with liquid chemical in between at the place where our beams cross. But how do we study changes over time? Because I said time. Time is a very important component of this. So we use a technique that was actually developed by the head of my lab, Dr. Kathy Wong. She developed this working with another grad student. And it's called SSTA, or single shot transient absorption. And it's very complicated, but don't worry, I'm going to break it down for you, and hopefully it'll be somewhat understandable. It all boils down to triangles. We use triangles. So here we have a triangle with this green side right here and this white side right here. Yeah. And at this point, the two lines are touching. There's absolutely no distance in between these two lines. But if we move a little farther out, then there's, say, 15 units in between these two lines. Move a little farther out, 30, and then all the way out there is 60. Does that make sense? Based on where we are in the triangle, there's different amounts of distance in between them, because triangles. So distance, in this case, is equal to time. So right here, if we send the beams in at an angle, then they cross at different times. So right here, they're hitting at exactly the same time, yeah, because they're overlapping. Right here, they're hitting 15 units apart. So in this case, our units are picoseconds, which is super, super, super small. And then we can also see, like over here, they hit 30 picoseconds apart, and over here, 60 picoseconds apart. And just to remind you again, this green beam is the pump beam, which is the one that sends the energy into sample. That's the poking it with a stick. And then this white beam is the probe beam, which is the one that gathers the information. So that's the looking at it and seeing how it responds. And the sample is down there. Okay, so why do we need this setup? I just mentioned that our setup works in picoseconds. Why do we need something that has that short of time? The reason why we need it is that the systems that we look at change really, really quickly. So these are nanocrystals at 1 minute. And so this is right after I've combined all of the different chemical ingredients into the little vial that we start the reaction in. I'll talk more about that later. And so this is after the reaction has been running for 1 minute. And this is after they've been running for 3 minutes. And that is a dramatic change. So if we're going to see what happens in between that change, if we're going to like track those changes, then we have to have something that works really quickly. And all of the other measurement techniques that exist are just way too slow. They take multiple hours versus the seconds that it takes us to get data. So nanocrystals, let's talk about nanocrystals. Why are we studying them, first of all? Nanocrystals are really interesting because they fluoresce or give off light at all colors. So you can see here, here's a picture of cesium, lead, chloride, bromine, chlorine, sorry. Yes, so cesium, lead nanocrystals, and you can see here's the red, and then it goes all the way over to purple. And we can use these nanocrystals to create more energy efficient LEDs. So over here, we have pictures of LEDs that not our lab, but a different lab built as kind of a proof of concept that these can work. And of course, if we can reach all different colors, then that means we can make LEDs of any color, and they're much more energy efficient. But what do nanocrystals look like? Nanocrystals, as the name implies, nano, are very, very, very small. So over here is a cartoon of a nanocrystal, and you can see kind of, there's like crystal lattice, as the name implies, and then there's these little squiggly molecules that stick off and act as kind of a shell that keeps the nanocrystal safe in the solution. And I said in the solution, we grow these as they're suspended inside of a liquid or a solvent. And so to give you an idea of how small these are, if a nanocrystal was the size of a football, then a single one of our red blood cells would be the size of a football field. So these things are super, super small. So making nanocrystals. How do we make nanocrystals? Or how do I make nanocrystals? Usually I'm the one in the lab who's doing the nanocrystals and actually creating them. So first of all, I weigh out the various ingredients. We call them precursors, just kind of the technical term, but ingredients also works. And so there are the solids and the liquids. In the solids, we have lead is a big component. We have a compound that contains iodine, also a big component of ours. And then, an organic molecule. And then in the liquids, we have our solvent. So remember, that's kind of the liquid that the reaction happens in, but the liquid itself isn't involved in the reaction. And then we also, in that liquid mixture, we also have what we call ligands. But you don't need to know that term, but that's the squiggly bits that are kind of sticking off and like protect and cap the nanocrystal. So that's also in the liquid mixture. And so we combine that all into a vial in the sonicator, which is a kind of machine that stirs solutions using water, a water bath, and high-pitched sound. And so it's pretty cool. I have a video of nanocrystals in the sonicator, but I don't have any audio for this video because the sonicator is so high-pitched that it really doesn't record well. But it's on like a little float that's in a water bath, and the high-pitched sound is running through it and stirring the reaction, making sure everything reacts evenly. And so once we combine it all in the vial and the sonicator, nanocrystals start forming and growing in the solution. Okay, and then once they're forming and growing, we need to measure them. So we use syringes to draw and then filter our samples. So you can see here, I'm drawing out a sample into the syringe from the nanocrystals, and you can see there's the vial, and then here's the little float that we use to keep the vials floating in the water bath. And so once I draw out that sample, I put on a syringe filter, which kind of acts as like, say, a coffee filter to get out all of the big particles. And so in this mixture, we've got particles of maybe unreacted lead, or unreacted things that haven't really done anything, and that can interfere with our measurements. So we want to get those out, we want to filter those out like a coffee filter. And so filtering them out, and then we put those into a cuvette, which again is those two thin layers of glass with liquid in between. And we draw these samples at different times during the reaction. So 3 minutes, 5 minutes, 10, 15, 20, 25, 30, all the way out to 120 minutes, which also 2 hours. And so we want to see how the reaction progresses over time. And so once we have the nanocrystals in the cuvette, then we use the laser to collect our measurements. So over here you can see a picture. This is the cuvette that we use with the laser. And you can't see the laser up here, but remember again, that's because you can only see the laser when it's bouncing off, say, particles in the air or dust or condensation. And in this picture, that's not happening. But you can see the laser when it's hitting on the cuvette. So that line right there is the laser. And so remember again, the laser uses the pump beam, which is sending the energy in, and that's poking it with a stick, yeah? And then the probe beam, which is the one that gathers the information, that's watching to see how it responds. And so we get data, we run the laser, and we get data. And then once we have that data, we need to look at it, because it's not useful unless we can like look at it and see how it happens and make some conclusions off it, right? And so that is when I use Python to plot up the data so we can visually see what's happening during the reaction. So over here is an example of my code. And then over here is an example of one of my plots. And you can see this is really useful because not only can we show other people what we're doing and the data that we've collected, but we can also see like, okay, here at 15 minutes, this very dark blue line, the peak, that is tallest around like 600 nanometers. But then all the way at 120 minutes, that is tallest around, I don't know, what is that, 660? About there. And so that we can make conclusions off of that based on what peaks have kind of gotten shorter or disappeared and what peaks have gotten taller. And yeah, and so we can make conclusions about how the reaction has progressed and how it's happened. Okay, so moving on from nanocrystals, I'm going to talk about organic thin films really briefly and what we're doing with them. So an organic thin film, I should probably define that first, is a very thin layer of chemicals on a surface. And so the surface that we use is like a glass microscope slide, as I was talking about earlier, but you can use lots of different surfaces. I've read about some labs that use plastic or other labs that are making electronics and use gold because gold conducts electricity really well. And so we make these films, which are the thin layer of chemicals on the glass slide. And then what we're studying is how heating the film changes the electronics. And so over here, you can see a picture of a film that is before annealing, or annealing is another term for heating it up. And so this is a film that before we've heated it and a film after we've heated it. And we want to know what happens on the super small scale while we're heating it. What changes about the film? Because we can see that there's been a change. And if we look at it like electronically, if we look how it performs electronically, we can also see that there's been a change, but we don't know why. And so we want to see what happens, how do the molecules change on the surface as we're heating it. And so the application for this is solar panels and flexible electronics. So solar panels are especially a big one. You can see over here a picture of a flexible solar panel. But so these chemicals that we're working right now, P3HT and PCBM, are kind of a model system for solar panels. And so if we can figure out what happens as we're heating it up during this check mark or during this question mark, then we can take that and apply it to all different materials that solar panels are made of and use that knowledge to make better solar panels and more energy efficient solar panels. So it's pretty cool. Yeah. Okay, conclusion. So I've talked about what we're doing, nanocrystals and then organic thin films, and I've talked about the laser, but what are my takeaways from working in the research lab? I've been working in here for a year, so like I've gotten time to learn kind of big takeaways. So my first takeaway is that there is so much cool research going on. Every week for lab, or for our, we call group meeting, we have to read a paper and then give a short kind of presentation blurb on it. And through that, I've gotten to read a lot of different papers. And it's fascinating to see how many different questions people are looking into and attacking in different ways, and all of the different techniques that people are using and trying and developing. Like the head of the lab developed her own technique to look at the thing, and that is going on all over, and that's super cool. So yeah, there's so much research going on everywhere. Another thing is that research is incredibly interdisciplinary. You might have picked up on it. I mean, I started this talk talking about a laser, which probably falls more into, I don't know, engineering or physics or, yeah, but it's so important to the work that we're doing. And so engineering and physics are just kind of a natural part of what we're doing. And also computer science, coding. I showed you the code that I wrote. I had to learn a lot of computer science and like Python in this lab because you need that to present your data or to study things. I have a I have a lab colleague whose entire PhD project is just to build a model of these systems with Python, which is super cool. Um, and then you also have other sciences coming in, like biology. For group meeting, for my paper that I've read for group meeting, I've read biology papers because they've been applicable to the stuff that I'm doing. And so there's so many different sciences incorporated in research and so many different skills incorporated in research. And so if you're interested in doing a lot of things, then research might be a good choice. Final big takeaway is that it's just really fun. It's fun to combine chemicals and to study how they react. It's fun to go after questions that nobody's ever answered and to look into what is happening at the fundamental level. And it's just really fun. Okay, so again, so if you're interested in the things that I was talking about, or you're interested in doing research, not just chemistry research, any kind of research, then I would highly suggest that you come to WISE meetings, or Women in Science and Engineering. Our meetings are Tuesday at 1 p.m. and Dr. Miller should have sent out, or did send out, a Canvas announcement with the link, with the Zoom link. And yeah, so if you're interested in any kind of science or STEM topic, we have chemistry, obviously, but we also have engineering or mathematics or biology, medical sciences, psychology, any kind of science or STEM subject. So if you're interested, please join us in the WISE meetings. Everybody's welcome. And if you're interested in potentially interning or volunteering in a research lab or shadowing somebody for a day, shadowing a research scientist for a day, then I would highly suggest coming to the meeting as well, or emailing our lovely advisor, Mary Beth, because she has connections at U of O research labs and many different fields, and might be able to set you up and connect you with a professor or a head of a lab somewhere. So if you're interested, come to the meeting. All right, and that's all I have. Any questions?